It was also possible to study how thousands of genes and proteins work together inside our cells. HGP itself was made possible by two major methodologies of biological research: expressed sequence tags, or ESTs, or DNA sequence tags and sequence annotation. In EST or DNA sequence tag method, the focus was on identifying and isolating all genes that expressed as RNA. Actually, EST is the term for all genes that express as RNA. With EST, numerous databases of nucleotide sequences have been made available. It has facilitated the construction of the preliminary transcript map of a human genome. In the second methodology, the entire set of genome that comprised all coding and non-coding sequences was indiscriminately sequenced, and later different regions of the sequence were assigned with a function. The assigning of functions to sequences is called sequence annotation. For the purpose of sequencing, the complete DNA was extracted from a cell and divided into smaller fragments, since it is technically difficult to sequence longer fragments of DNA. These DNA fragments were then cloned in suitable hosts such as bacteria or yeasts with the help of specialized plasmids or vectors. The vectors were called bacterial artificial chromosomes or BAC and yeast artificial chromosomes or YAC respectively. The cloning amplified each DNA sequence, which made it easier to sequence. Sequencing was then done by automated sequencers that worked on a principle developed by Frederick Sanger, who was also responsible for developing a method to sequence proteins and amino acids. As per Sanger's method, the cloned DNA sequences were arranged based on the overlapping regions present. For this, overlapping segments were generated. However, aligning this was not an easy task. As per Sanger's method, the cloned DNA sequences were arranged based on the overlapping regions present. For this, overlapping segments were generated. However, aligning this was not an easy task. Therefore, specialized computer-based programs were developed that annotated these sequences and assigned to a chromosome. Out of the 22 autosomes and 2 allosomes X and Y, chromosome 1 was the last that has been sequenced in May 2006. Did you know that chromosome 1 has the most number of genes? That is 2,968, whereas chromosome Y has the least number of genes, only 231. These challenging tasks would not have been possible if advanced genetic engineering methodologies had not been put forth by the HGP, consequently paving the way for useful biological research. In the past, it was believed that proteins were the genetic material. However, in time, through the various experiments using bacteria and viruses, the truth about genetic material came to light. British bacteriologist Frederick Griffith was the first to conduct experiments with bacteria Streptococcus pneumoniae that was responsible for causing the disease pneumonia. He worked with two types of bacteria strains, the pathogenic S strain and the non-pathogenic R strain. The S strain has a polysaccharide coat that makes it smooth, whereas the R strain lacks it, making it rough. During his experiments, Griffith realized that when heat-killed S-strain cells were mixed with R-strain living bacteria, and this mixture was injected into healthy mice, they died of pneumonia. Thereafter, when the bacteria were isolated from dead mice, they were found to be of the living S-strain type. This proved that the R-strain bacteria had transformed to the living S-strain. Yet for a long time, due to lack of conclusive proof, proteins were believed to have caused this transformation. However, in 1944, Oswald Avery, Colin MacLeod, and Macklin McCarty worked together to determine the biochemical that had caused the transformation in Griffith's experiment. They repeated Griffith's experiments and purified all three biochemicals such as proteins, DNA and RNA from heat-killed S-strain cells to identify which one had caused the transformation. They discovered that neither the protein-digesting enzymes, that is, proteases, nor the RNA-digesting enzymes, that is, RNases, affected this transformation. Whereas DNA-digestive enzyme, DNase, did inhibit the transformation. Therefore, it was clear that the transforming substance was not protein or RNA, it was DNA. 8 years later, in 1952, the definitive evidence that DNA was a genetic material was provided by Alfred Hershey and Martha Chase. They conducted their experiments with viruses that attack bacteria, better known as bacteriophages. Bacteriophages attach to bacteria and implant their viral genetic material inside the bacterial cell. The bacteria treat this genetic material as their own and produce more of such virus particles. Since Hershey and Chase wanted to find out whether it was protein or DNA from the virus that entered the bacteria, they grew these viruses on two different mediums, one with radioactive phosphorus and the other with radioactive sulfur. As DNA contains phosphorus while protein doesn't, the viruses that were grown in the presence of radioactive phosphorus had radioactive DNA and not radioactive proteins. On a similar note, as DNA doesn't have sulfur like proteins, the viruses grown on radioactive sulfur had radioactive proteins and not radioactive DNA. Thereafter, both these radioactive phages were allowed to infect E. coli bacteria. Later, after the infection, when viral coats were removed from the bacteria, it was revealed that the bacteria that were infected with viruses containing radioactive proteins were not radioactive. On the other hand, the bacteria that were infected by viruses with radioactive DNA were radioactive. It was clear then that proteins did not enter the bacteria from the viruses. Rather, it was the DNA that passed on from the virus to the bacteria. Therefore, despite experiments by Griffith and Avery, MacLeod and McCarty, it was the experiment by Hershey and Chase that proved the fact that genetic material is DNA. Did you know that except for identical twins, it is very unlikely for two individuals to have the same DNA pattern? It is the small difference in base pair sequences of DNA that make the phenotypic appearance of each individual unique. The human genome has about 3 billion base pairs. Therefore, it would be a time-consuming and expensive task to find out the genetic difference between two individuals, as it would require comparing two sets of 3 billion base pairs. An easier and quicker solution to comparing DNA sequences is DNA fingerprinting, which is a test to classify and analyze DNA or the genetic information. In human beings, 99% of DNA base sequences are identical and are known as the bulk genomic DNA. The remaining 1% DNA base sequences differ and are present as a small stretch of repeated sequences known as repetitive DNA. DNA fingerprinting identifies the differences in this region. To separate both genomic as well as repetitive DNA, the process of density gradient centrifugation is carried out. As satellite DNA is lighter and bulk DNA is heavier, they get separated on the basis of their density. Graphical representation shows bulk genomic DNA as a major peak and repetitive DNA as smaller peaks known as satellite DNA. Satellite DNA is highly repetitive and consists of non-coding sequences. Based on the length of the segment, base composition, and number of repetitive units, satellite DNA can be classified as mini-satellite DNA and microsatellite DNA. Mini-satellite is a section of DNA which has a variable number of tandem repeats, or VNTR. It has sequences of 15 to 100 base pairs repeated hundreds or thousands of times. On the other hand, microsatellite is a section of DNA which has tandem repeats of shorter sequences of 2 to 10 base pairs. Both mini-satellite and microsatellite DNA act as molecular markers in the DNA fingerprinting technique. These satellite DNA do not show any impact on reproducing ability and exhibit a high degree of DNA polymorphism or genetic variations within a population. DNA polymorphism is the result of mutations in either somatic cells or germ cells, which accumulates over generations. DNA polymorphism is the guiding principle behind genetic mapping and therefore helps in the DNA fingerprinting technique. The DNA fingerprinting technique was developed by Alec Jeffreys. This technique involved southern blot hybridization, which used radio-labeled VNTR as a probe. VNTR is a small sequence of DNA arranged tandemly in several copy numbers, which varies from one chromosome to another in an individual. A very high degree of polymorphism is seen in the number of these repeats, due to which the size of the VNTR varies from 0.1 to 20 kilobytes. Let's now study the steps involved in the DNA fingerprinting technique. First, the DNA is isolated and then digested by restriction endonucleases, which results in DNA fragmentation. Next, these DNA fragments are separated with the help of gel electrophoresis and then transferred to synthetic membranes like nylon or nitrocellulose. This step is followed by the hybridization of the DNA fragments using a radio-labeled VNTR probe. Finally, the hybridized DNA fragments are detected by a technique called autoradiography, conducted using an X-ray film. Hybridization with the VNTR probe results in an autoradiogram, which produces several bands of different sizes. These bands provide a characteristic pattern to an individual's DNA and vary from one individual to another except in identical or monozygotic twins. Today, the accuracy of the DNA fingerprinting technique has further improved due to the advent of the polymerase chain reaction, or PCR, where multiple copies of a single DNA sequence can be made. DNA fingerprinting has also advanced due to the use of different types of DNA probes. DNA fingerprinting is used in many areas. It utilizes DNA as an identification tool and helps in forensic applications such as crime investigation. It also helps determine genetic and population diversity, forms the basis of paternity testing in case of parentage disputes, and also helps in the study of evolution and speciation. In this way, DNA fingerprinting has revolutionized the process of DNA testing, as it can identify the minute differences between various DNA samples. The human cell is an amazing structure which is busy carrying out various life-sustaining processes, one of which is protein synthesis. For this process, it is essential that amino acids which lie scattered in the cytoplasm are carried to the messenger RNA or the mRNA template, the site of protein synthesis. Till the 1950s, scientists did not know enough about how proteins were carried to the mRNA. The breakthrough came in 1955, when Francis Crick, who had earlier revealed the DNA structure and its properties, postulated the adapter hypothesis theory, in which he stated that it is therefore a natural hypothesis that the amino acid is carried to the template by an adapter molecule, and that the adapter is the part which actually fits onto the RNA. In its simplest form, this hypothesis would require 20 adapters, one for each amino acid. Soon after Crick published his hypothesis, scientists across the world worked to find this mysterious adapter molecule that adapts itself to carrying amino acid and reading it to the codon of mRNA. Success, however, came to Robert Hawley when in 1965 he discovered transfer RNA or tRNA, the adapter molecule, and also worked out its entire chemical structure. Today we know that tRNA, the smallest of all RNA molecules, is made up of about 75 nucleotides. Synthesized from a DNA molecule in a process similar to the synthesis of mRNA, tRNA is soluble in 1 molar NaCl and is hence known as soluble RNA or sRNA. Each tRNA has G at its 5' end and CCA at its 3' end. Interestingly, tRNA takes the shape of a clover leaf when examined as a two-dimensional structure, whereas it looks like an inverted L-shaped molecule in a three-dimensional structure. Regardless of its dimensions, a tRNA consists of 4 arms or sites: ribosome recognition site, anticodon site, enzyme recognition site, and amino acid binding site or carrier end. Each of the tRNA regions perform a particular function. Take the case of the ribosome recognition site, which recognizes specific ribosomes attached to the mRNA and brings amino acids to the ribosome for protein synthesis. On the other hand, the anticodon site consists of 3 nitrogen bases called the anticodon. During protein synthesis, the anticodon pairs with the codon of the mRNA strand. However, the anticodon of tRNA is complementary to the codon of the mRNA strand. Apart from the anticodon site, a tRNA molecule also consists of an enzyme recognition site, also known as the D-arm or dihydrouracil loop. This particular site is the recognition site for amino acid synthetase enzyme, which helps the amino acid bind to the tRNA. Incidentally, this binding takes place at the 3' end of the amino acid binding site. Did you know that there is a specific tRNA for each of the 20 amino acids found in the human body? Also, there is a specific tRNA called initiator tRNA, which initiates the process of protein synthesis. However, there are no tRNAs for stop codons. tRNA is thus an important molecule, and it plays a vital role in protein synthesis by bringing amino acids to the mRNA template in the cytoplasm.